you, but we
Oh,
can
yes,
see
I'm
the
doing.
share screen.
Okay. How do I? Hold on. Let me see if I can. Oh, oh, that's why. Now?
Okay, now we can also see you.
Okay, good. I'll share the screen again.
So when you're ready, please start. Thank
Okay.
you.
Okay. Thank you so much for the kind introduction, Pascal, and also for letting me speak at this session at this conference. First time here, which is very exciting after seeing very nice talks yesterday and today. Um, so I'm a postdoctoral researcher at the Numerical Simulation Lab at the University of Saskatchewan, and I'm going to talk about essentially what my title is trying to say is that I'm trying to compare these three models in the context of cardiac electrophysiology, right? Um, so here's a little outline. I'm going to talk about the well-known mono and bidomain models, then the Emmy model, which is a fairly new model used in cardiac physiology nowadays, and then some numerical methods. Then I'll show a few comparisons that we have, so through some preliminary results, and then I'm going to finish with some conclusions, ongoing work, and future work. Okay. So first, I like to acknowledge my team who make which makes my work possible in some way through our collaboration. Um, the first one is Raymond Spiteri, my supervisor, who is a professor at the University of Saskatchewan in the computer science department. Also, my colleague and collaborator Kevin Green, who is a research scientist in the same department at the same university, and also Joyce Reamer, who is a master's student in our lab, but who is officially part of the division of biomedical engineer at the same university. So I'm going to start my talk by telling you a little bit about, well, the well-known monodomain and bidomain models, which probably I don't need to talk about too much um, for this audience. But essentially, I have them in the order of monodomain first and bidomain, but usually we think about the monodomain as a special case of the bidomain model, right, where the extracellular conductivity is a multiple of the intracellular one, right? So for me, or for this in this talk, this V will mean the transmembrane potential, whereas the UE is the uh, extracellular potential. Um, a fairly large number of papers have been published around these two models and the, regarding many different contexts. We're particularly interested in the ones that are referring to the discretization, the methods that are used to approximate the solutions to these problems. Um, I have two example sample references with Serbian Spiteri in 2018, who worked with the bidomain and monodomain models in 1 and 3D and showed that higher order operator splitting methods are more efficient than people thought before for these problems. And also Chamakuri in 2019, who developed a space-time adaptive method, which works in parallel. And I put these two references here because these are related to the work, in some part of the work that we're doing for these two models, plus the next model that I'm going to present, which is the Emmy or EMI model, which is a newly developed model in the context of cardiac physiology, but it's also used in other applications such as neurons. Um, the Emmy stands for extracellular, membrane, and intracellular. In this model, similar, or actually, it looks like a mono or bidomain actually a model, but in this case, we have actually membranes of cells being explicitly defined in our equations. So this means that now, as I have here in this picture, I I have two cells that are represented by this omega I1 domain, and the cell is called gamma 1. So that's the cell membrane of for the cell omega I1. And I have another cell here, which are joined through one of their faces. And I have an extracellular space omega. So that's why, well, that's the kind of the detail of the domains that we consider in this Emmy model. And this this place or this phase where the two cells are touching each other, right, or are thought of modeling gap junctions in cells. Okay, so the idea of this model is to represent electrical activity at the cellular level. So we can have the idea is to represent the intracellular potential and the extracellular, and also the transmembrane potential on the membrane cell. And it may be able to address some limitations of current neuron and cardiac models, which due to its more detailed description, we may able to, for example, generate geometries for cells, which are more or less unique. And also, we could specify heterogeneous ion channel distribution on different points. First work works towards the use of such cellular level models are by Stienstra in 2009, where he studied something he called microdomain model to compare conduction velocity with bidomain. And also the model that I'll show you today, and the one that we actually work with, is it was first released by Agudelo, Toro, and Nif in 2013. Okay. um, and also the things that I showed you today are actually what we base our implementation on is by what was done by Tevedo et al. In 2019. Okay. So the Emmy model looks like this. So we have some diffusion equation, not parabolic anymore as in the bidomain or monodomain, but a simple static diffusion equation in both the extracellular domain. For example, if you have one cell, and then in the intracellular domain, you have another diffusion equation. This K superscript K means that you may have more than one cell. For example, this domain now, we have all the two cells instead of one. And so, and we have cell equations that actually work on the membrane of these cells. So between the intra and extracellular domains, we have called the omega IK to that part of the cell membrane, and we have the cell equations, cell model equations that which describe the dynamics on of the potential, action potential on the membranes, and also we have a similar model on the gap junctions that we call gamma 1, 2. Actually, this is a typo here. It should be DOMINGUEZ actually. Also on this membranes, we have that the transmembrane potentials are defined as the jump across the membrane, right, of the action potentials, right? We also have some boundary and flux conditions. So the first one on the outer boundary, we assume that the simple Neumann boundary condition. So it'll be flux free. And then on the interfaces, we have that the Neumann data is continuous. So we have a continuity in the normal derivative for that. And we also define these currents that we had in the cell models from before, are right here, right? As this flux of potential across the membrane. On the gap junctions or between the cells, we assume a passive model for the current gap, and it's simply 1 over the resistance times the potential on the membrane, right? And of course, this is coupled with the cell model and the intra and extracellular membrane, which looks like this, right? This is, as usual, this is dependent on the cell model that we use for our implementation or study, okay? So for what I'll show you today, we use special discretization using a mixed finite element where we compute the flux of the action potential, the action potentials, and the transmembrane potentials, and which is implemented in the FreeFEM++ library. Today, I'm going to show you preliminary results which are done or computed on this simple domain with 2 cells and 1 gap junction. In the time discretization, we use a first order Goodenough operator splitting, which looks like this. After we have discretized the space with the mixed finite element formulation, we consider the new ODE that looks like this, and then we have an explicit part which is taking care of the cell model, and then the implicit part which is taking care of the tissue model. Okay, we use a forward Euler for the cell model with time substeps to resolve the dynamics correctly, or as correctly as possible, of course, and a backward Euler method for the tissue model. Right? So some recent work that we have done with the EMI model was to study LQT1 syndrome and the effects of it, and how we could use the Emmy model or EMI model to actually to see the effects of LQT1 syndrome. There we showed that the Emmy model was able to predict the behavior that seen in experimental data within some reasonable range. I'm not going to go too deep into this, but I'll point you to a talk tomorrow by my colleague Joyce Rimmer, who will be giving a lot more details on LQT1 syndrome and Emmy model. Right? Please consider that talk. So about the tuning parameters, or actually comparing these 3 models, they do look that it looks like some of them, or some of the parameters look the same in terms of the PDE model, right? But we see that, for example, as we already know for the mono and the bidomain, that some of the parameters are not present in one model or the other one. And for the Emmy, we also have that. And in particular, the most important one is probably the resistance at the gap junctions. And also the fact that even though we wrote it in the same way, the conductivities may not mean the same thing due to the way that the models are written. So we wanted to answer essentially 2 questions about this Emmy model. The first one is, how different is the output from the mono, bi, and Emmy models? Are also what parameters need to be tuned, or how we can tune those to get similar outputs from these models. All right. Because as first look, if you look at the equations, you may obviously think that you may not get the same output, since the dynamics work in a different way, right? So here I did a simple test to see how the APD90 would behave, or the actual potential actually would behave over time. And so by taking some parameters from other papers, in particular for the mono and bidomain, I took some parameters using there from Servi and Spiteri in 2018. And also, it also took some parameters for the Emmy model from Tibeto 2017. And I used the 10-to-share and Panfield cell model to run my simulation and see what would happen. And then after running the simulation with these parameters, I wanted to see how much the parameters would change if I wanted to get a similar APD90. So here I have in this plot, I have the action potential uh, profile of the 3 different models with the parameters that I showed before, and you can see that they recover differently. So I wanted to see how much we needed to change the parameters. So I ran some optimizations to tune the parameters and tested against some control data from Tentuscher et al. 2006, and set the tolerances to 10 to minus 4. I found out that if I varied, if I wanted to optimize for the conductivity in the monodomain and the lambda parameter, and in the bidomain for the 2 conductivities, and the Emmy only the resistance and the gap junctions, you will be able to actually see a much closer match for the APD90, even though the monodomain and the bidomain took a lot more iterations to converge to the tolerances that we set um, than before. Okay, so some ongoing work that actually involves the comparison of these 3 models are also the implementation of our cardiac physiology models in DL2 and FreeFrame and the FreeFrame libraries. Also, we are thinking about parallelizing our implementations with the P4S library for meshes and the Trulinos and PETSI linear solvers. And after seeing the talks today, we had very nice ideas on what we could add to our implementations. We're also thinking about general time stepping and operator splitting methods in DL2 to be able to couple these with the models that we have implemented. Lastly, we are looking into full space-time adaptivity with sundials and DL2, right? Some conclusions is that the parameters can be tuned for different models to obtain the results that we wanted to see. We are able to match the output from the monodomain, bidomain, and EMI models to experimental data. Some future work on actually ongoing work as well is that we want to use other cell models as well, and maybe even tune cell models, parameters in the cell models to see to see similar outputs, and also of course continue to work on this comparison. With that, I'll thank you, and I'll take questions.
Okay, thank you very much for the presentation. There is room for questions, so if anybody. Okay, there's the raised hand. Joshua, please go ahead.
Yes, hello. Can you hear me?
Yeah, but I can't see who is speaking. Sorry.
Oh.
Let me, oh, there. Okay, thank you.
Okay. Hey, how's it going? I have a question. So first of all, thank you for the talk. It was very interesting. It was very insightful. And it was really nice to learn about the EMI model. I wasn't aware of it. I was wondering if you expect to have similar results when you include BATH to the bidomain model. You know, like, because I'm guessing that that could bring some other, maybe changes to, the discretization, or I don't know. I was just wondering what your thoughts were about, including BATH to the bidomain.
So yeah, thanks for the question, Josue. That would be something we will have to look into, because now, like, even for the plain model where we only have the electrophysiology models, right, we see differences from the models, right, or especially the EMI compared to the other 2. But yeah, like if you add more conditions or more equations to it, right, we would expect to see differences as well.
That makes sense. Thank you.
Yeah.
Okay, other questions? Actually, I have one concerning, at some point you mentioned about some optimization algorithm to tune the parameters among the different models. Did you use a specific optimization method, or was it just a trial and error?
So I'm simply running this stuff on MATLAB with the FME search, which uses I always forget the name. So let me go back to my slides. It's the Nelder-Mead simplex algorithm, right? So I wanted to use something that wasn't dependent on a gradient for this study.
Okay, thank you.
Yeah. Yeah.
Okay, I don't see other questions from the audience. This was the last talk for this session. I say thanks again to all the speakers and to all the participants